We have already discussed about the male reproductive part of a flower, and we looked deep down into how the anther looks, what is inside the anther, and the detailed cross-sectional structure of an anther. And the main purpose of that video was to see how the microspores are formed here. So let me bring this part outside. So this is the one which has sporogenous cells. These pink cells that you see inside are sporogenous cells, and we saw how these sporogenous cells divide meiotically to form microspores. Now in this video we will look into what are the changes that takes place inside these microspores so that a pollen is formed, and then how this anther bursts and pollens are released. So let's begin. Let's begin by talking about the changes that takes place in the microspores, and for that we'll have to look into this part. So the tapetum layer and the pink cells that you see inside, which later forms microspore. Let us enlarge that part now. So here it is. This is the tapetum layer. Let me write down. This is tapetum. And why is it so important? Because the cells here are big, it has a lot of cytoplasm, and it provides nourishment to the cells that's growing inside. Along with that, it has many other functions, and we will discuss about them when we come across those functions, alright? So inside this tapetal layer, we will have microspores, which are formed by meiotic division, and therefore they are haploid, and we discussed how these cells are formed in our previous videos already. Okay, now we will look into the changes that takes place in the microspores, and for that, let us just keep one cell and look into the changes that's taking place in that cell, okay? So let's get rid of all these cells and just keep one. Here it is. This is also a haploid microspore. Now this microspore has a membrane which is just like a cell's membrane. So it is similar to plasma membrane. But around this plasma membrane, this microspore now starts secreting another layer which is called the intine. This intine is made of cellulose and cellulose plus pectin. Okay, so these are the substances that makes up this layer, and who produces this intine? This microspore itself. Now, what is the need of this microspore to even produce a layer outside it, because it already has a plasma membrane, right? So what is the need of another layer? What do you think? Well, the answer is this microspore, which later will turn into a pollen now, will be released outside in the environment to go and fuse with the female gamete, right? So to protect the content inside the microspore, it needs layers outside so that it is not destroyed when it is released in the environment. Now, this tapetum layer, which is outside, which provides nourishment to the growing cells inside, this tapetum layer thinks that this intine layer is not sufficient for the microspores. So, this tapetum layer now secretes a substance which is called a sporopollenin. This sporopollenin aggregates around this microspore, producing another layer which we call exine. So this exine is secreted by this tapetum layer. And this sporopollenin, this long biological term, is actually a chemical substance which is one of the most resistant organic compounds known. And this sporopollenin makes up the exine. So imagine the kind of resistance or the kind of protection this microspore or the pollen gets. So if an animal happened to ingest this pollen, this exine will make sure that the digestive juices of this animal won't digest the pollen. And also, if this pollen happened to fall on parts where it should not fall, like on water or on soil, then this exine layer will make sure that this pollen is not degraded or destroyed. So these two layers are basically to protect this pollen when it is released outside. And now, with two layers on the outside of this microspore is now called a pollen. We have already been calling it a pollen, but any microspore that has an intine and an exine outside it, we call it a pollen. So now it has become a proper pollen. Alright, now as we have discussed about the different layers that is outside a pollen, it's now time we look what's happening inside. So this pollen, which is formed from a microspore, is also haploid because microspores were haploid and we have not seen any cell division here. So this will also be haploid. But now mitosis will take place inside the cell. And like any mitotic division, first, the nucleus will divide. So the nucleus will divide into two. But things happen differently when it comes to the cytoplasmic division because the cytoplasmic division is not equal. As you can see, a large cell is formed which has more cytoplasm and more cellular content, and a small cellular section is formed which gradually forms a complete cell that floats inside this large cell. Now as we can see that this cell is divided into two, one large cell and a small cell. This large cell is now called the vegetative or the tube cell. Can you guess why we call it a tube cell? Well, this is because this cell gives rise to what we call a pollen tube. So if we consider this to be a pollen and here are the eggs of a flower, then from this part of the cell, pollen tube develops. So this is the pollen tube that reaches the egg, and through this tube the male gametophyte travels. So this is the cell, the tube cell, which gives rise to this yellow color long pollen tube that you see here. And we also call it vegetative because it do not directly take part in the fusion, or it do not give rise to the male gametophyte. So this vegetative or the tube cell we just saw gave rise to a long pollen tube. Then what is the job of this smaller cell here? This cell, we call it the generative cell because this cell gives rise to the male gametes. And most pollens are released in this stage. This two-cell stage, one which we call vegetative or tube cell, and the other one which we call generative cell that gives rise to the male gametophyte. So in this stage, the pollens are released. Now let us see how these pollens are actually released. And for that, let us bring in the transverse section of the anther again. And as we have discussed, this pink sporogenous cells inside finally grows into proper pollen, and we saw how pollens are finally formed. But during the course of the formation of pollens, the layers outside the pollen particles slowly becomes fibrous, and somewhere here and here on both sides, the tension kind of increases and the anther bursts out, releasing the pollen. And that is how we see the pollen being released outside by the anthers. So this was all about how pollens are formed from microspores and how the anther bursts and releases the pollen outside.